So in a previous lesson, we learned how the DNA molecule can be duplicated. The secret to that process is the pairing rules. The T always pairs with A and C always pairs with G. So when the two strands of the molecule unzip and they separate, each strand can be used as a template to build the missing strand. In this lesson, we'll look at the second feature of DNA, how it can store information. And here, we're not going to be focusing on the stickiness, the pairing rules of the letters, but rather the sequence of DNA letters along one strand. So we'll be learning how that sequence of letters can be the recipe for a protein. To begin, we should consider what a protein is. It is a chain molecule composed of amino acids linked together in a chain. A little model's up here, the popcorn string here. Each popcorn kernel would be a different amino acid. There are 20 different amino acids. Now, amino acids are molecules like this shown in the picture. They're made of carbon atoms and nitrogen and oxygen and hydrogen. And one part of the molecule varies in 20 different ways. So there are 20 different kinds of amino acids that make up proteins for cells on Earth. Now, the chain of amino acids can be a short chain or a very long chain. If it's a longer chain, it will tend to fold up into a three-dimensional shape, and that shape determines the protein's function. So here we see the colored shapes will represent amino acids. We have a certain sequence of amino acids, and that defines a unique protein. It will fold up into a certain shape, and that unique shape will determine its unique function. So every protein has a job to do in the cell, and proteins are different because they have different sequences of amino acids. So the amino acid sequence determines the shape, and the shape determines the function or the job of the protein. If we have two proteins, protein 1 and protein 2, if they have a different sequence of amino acids, well, then they're going to fold up in different ways, and that's what gives rise to their different functions. So microtubule proteins have a different function than chopper proteins or receptor proteins. They all have different functions because they all have different shapes. They have different shapes because the sequence of amino acids is different. Now, in understanding how a sequence of DNA can code for a protein, it's instructive to notice that proteins in DNA are chain molecules. They're all built from a set of molecules put together in a certain sequence. So the possibility arose that the sequence of DNA letters might somehow stand for the sequence of amino acids in a protein. This was the big question facing biologists in the middle of last century. Could the sequence of DNA letters code for the sequence of amino acids? Here we'll take a look at some double-stranded DNA here. And again, we're not concerned about the pairing of the complementary letters here, but rather just the sequence along one strand. So the question on the table was, how can this sequence of letters that appears to be random to us, how can it be the recipe to build a protein? How could that sequence of DNA letters somehow code for the sequence of amino acids in a protein? Now notice there are just 4 different letters in DNA, the A, G, C, and T, yet there are 20 amino acids, so we're going to have to figure out how this sequence of 4 letters that differ can stand for 20 different objects, the amino acids. Well, a 1-letter code won't work. With only 4 different DNA letters, there could only be 4 amino acids, but there are 20 different amino acids in proteins. So for example, let's say the gene, the recipe starts here with this T. If T specifies a certain amino acid, so T would mean that a certain amino acid is in the first position of the protein, then the next letter would indicate that the C indicates that a certain other amino acid is here, and then the A means that the third amino acid has a certain identity. But since we only have 4 letters, every time there's a T, so there's a T down here, it would have to be the same amino acid. But wait, there are 20 different amino acids, so we can't have a code where 1 letter of DNA stands for a given amino acid. We just don't have enough DNA letters. We only have 4, and yet we have 20 amino acids to code for. What about a 2-letter code? Could it be that the 2 letters here, T and C, specifies the first amino acid? Then the next 2 letters, A and G, specifies the second amino acid. Then A and T specifies the third. Could that code work? Would we have enough two-letter doublets, so to speak, to stand for 20 different amino acids? Well, we can do a little mathematical reasoning here. How many two-letter DNA permutations are there? Now, since we care about the order, it's a permutation, not a combination. Well, in the first position, there are 4 possible DNA letters that can go in the first position. It could be an A and a T or G or a C. And likewise for the second. When you multiply them together, you get 16 possible two-letter DNA permutations. Here are some examples down here. Well, 16 two-letter permutations, but we have 20 amino acids. It's looking like that's not quite enough. And so a 3-letter code. 4 times 4 times 4 is going to give us 64 different DNA permutations. So there are 64 DNA triplets, and here are some examples. Now that would be plenty to specify 20 different amino acids. So this is our current understanding of how the DNA code works. It is DNA triplets that are like words in the language of biology. So 3 letters codes for an amino acid. So the first 3 letters in the sequence here specifies the identity of the first amino acid. The next 3 letters specifies the identity of the next amino acid, and so on down the sequence. Now, in the 1950s and 60s, there were lots of biologists who were sort of unhappy with this idea that there were 64 possible DNA triplets and only 20 amino acids. And so they thought about ways in which some of the DNA triplets wouldn't code for anything. They wanted to find a way that maybe there were just 20 DNA triplets that specified the 20 amino acids. But today we recognize that there is simply redundancy in this coding process. So more than one DNA triplet can code for the same amino acid. We have 64 DNA triplets, but only 20 amino acids. So each amino acid can have several names, in a sense. Okay, let's return now to how a cell is going to be making a protein. So here we are in the nucleus, and we have a section of DNA that appears to be opened up, and we have a messenger RNA molecule being made. And recall, we learned that messenger RNA was a temporary copy of the information in DNA. Well, it might not be a surprise to you to learn that messenger RNA is also a chain molecule. Its sequence is a copy of the DNA sequence. So we've got 3 chain molecules running the show for cells on Earth. We've got a sequence of DNA will be copied into a sequence of RNA molecules, and then that sequence of RNA is going to specify the sequence of amino acids of a protein. Let's take a look at how messenger RNA is built. At the top, notice we have a section of DNA that has been unwrapped, uncoiled from the histone proteins. You have to uncoil your DNA in order to be able to use the information. And the reason is we have to be able to make a messenger RNA from a stretch of DNA here. In the bottom picture, we see how this happens. The DNA strands separate, much like they did during DNA duplication, but this is when the cell's not duplicating DNA here. It's using the DNA information in order to build a protein. Notice one strand of DNA is going to be used as a template. Now, in DNA duplication, both strands were used as templates, but for messenger RNA construction, only one strand of DNA is used. So the process then of construction of messenger RNA is the first step in building a protein. Now in the nucleus, messenger RNA letters are floating around. And notice we see a lot of the same letters as we saw in DNA. We have G's and A's and C's. Oh, but now there is a difference. There's a U instead of a T. Messenger RNA does not have a T, it has a U. So as one strand of DNA is a template, so it's going to be sticky with its corresponding messenger RNA letter, we can start to see the pairing rules will come into effect here. So for example, if there's a G in DNA, there's going to be a C in RNA. If there's a G here, there's a C. Now here's the difference. If there's an A in DNA, there's going to be a U in RNA. If there's a C in DNA, there's a G. If there's a T, there's an A in messenger RNA. So notice when you look at this section of the molecule here, all of the pairing rules apply as in DNA with one exception. The exception is RNA has a U instead of a T. All right, so We have to imagine in the nucleus there are messenger RNA letters, individual letters, bouncing around, floating around, just as there are DNA letters when it's time for DNA duplication. But here these messenger RNA letters will find their sticky partners when the two strands of DNA have separated. One of the strands, the template strand, will be used to construct the complementary messenger RNA molecule. And then this molecule will detach and then leave the nucleus. Now let's use this picture to take a look at one of the key protein players that facilitates this process. So here we have a chromosome, and usually when people draw chromosomes, they make them the duplicated ones. So here we have a duplicated chromosome still attached here, but now they're unraveling some of the DNA. And here we have a section of DNA that has opened up, so this is our gene, and we have messenger RNA being built from one of the strands of DNA. And here's our protein. It's called RNA polymerase. This is the molecule that links together, chemically bonds together the individual letters of messenger RNA into a chain molecule. And that chain molecule is going to preserve the sequence of DNA. Notice the messenger RNA is complementary to the template strand, but it's identical to the other strand. So you see here, we would say this top one here would be the template strand, and so the messenger RNA is complementary. So we have the pairing rules here. But if you look at the sequence of messenger RNA here, it's identical to the sequence of DNA on the other strand, with the exception of the U. So we have GAC, we have GAC, GACU, GACT. So this is what we mean when we say that a messenger RNA molecule is a copy of the information stored in DNA. The information takes the form of the sequence of DNA letters. And so let's zoom out. Now we have our messenger RNA molecule. It is detaching from the DNA, and it's going to leave the nucleus. This is Hammerling's molecule, and it's going to find a protein construction factory, and it's called a ribosome. And it's here where the ribosome will put together the proper amino acids in the sequence dictated by the messenger RNA molecule. Now, protein construction occurs during interphase of the cell cycle. So with the DNA uncoiled, genes are accessible, and so their information can be used to make the proteins. You'll remember that if the cell's going to reproduce, it will have to duplicate its chromosomes, and then the chromosomes will coil up. Whenever you coil up the DNA, the genes are not accessible. So only when the DNA is unwound and unwrapped and can be opened up for the purposes of making messenger RNA can the information stored in DNA be accessed. Now in this final part of the lesson, we'll learn about the genetic code. And this is it. This is the language that life on Earth uses. Now you'll notice it's written in the language of messenger RNA. See, we see some U's here. And that's because, well, messenger RNA is the actual, the molecule that goes to the factory. So oftentimes you see the genetic code written in the language of messenger RNA. Now, let's see how to use this tool here. Well, recall, messenger RNA then is going to be the molecule that is preserving the sequence stored in DNA. So we're going to think about the messenger RNA as a series of triplets. So a bunch of triplets in messenger RNA are going to direct the ribosome to put amino acids in a certain order. So the way you use this chart is that the inside ring shows the 4 possible messenger RNA letters that can be in the first position of a triplet. So you can have an A, a C, or a U, or a G. Could be the first letter of a triplet. The middle ring shows the second position, and the outer ring shows the third. So here's how it will work. Let's take the first triplet of the messenger RNA molecule, AUG. First position is A. So I go in the inner ring and I look for the A quadrant. Here it is down here. Second letter is a U. So within the A quadrant, I find the U section, the middle ring. Third letter is G. So within the A quadrant, in the middle ring U here, I go to the outer ring and I find the G, and that tells me that the amino acid is methionine. The abbreviation is MET. So the first amino acid in our protein is methionine. The second 3 letters, CCU, we can decode that by finding inside the inner ring. I find the C quadrant, and then the second was also a C, so C and then C, and the third was a U, so C, C, U. And that puts me in a section for proline. So the second amino acid is going to be proline. Notice, the triplet could have been CCU, it could have been CCC, CCA, CCG. All four of those triplets would code for proline. This is the redundancy that we learned about before. The third triplet is going to be GAC, so we go to the G, then the A, and then the C, and that's aspartate. Fourth triplet is going to be GUA, G, and then a U, and then an A, and that's valine. Now, all messenger RNA molecules have a stop at the end. Now, what is a stop? Well, you'll notice these three circles here. There are three triplets that tell the cell machinery that the messenger RNA molecule is complete. It's kind of like punctuation. So the last triplet of every messenger RNA molecule will be one of these three stop codes. So it would be UGA, or it would be a UAA, or a UAG. So every messenger RNA molecule will have a stop triplet at the end, and that tells RNA polymerase that the message is done. And so we have arrived at our final understanding of the language of biology. The language of biology is that a certain set of triplets of DNA letters are like words in the language of biology. And each DNA triplet copied in messenger RNA specifies one of the 20 different amino acids that all proteins are constructed. So the sequence of DNA copied as a sequence of messenger RNA will be translated into a sequence of amino acids that then folds up into protein. Every single protein in a cell has a unique sequence. Every different protein has a unique sequence stored in its own gene. So chromosomes are long pieces of DNA, and they carry hundreds of recipes, hundreds of genes for the thousands of proteins that cells need to make in order to stay alive. And we'll leave with just one last look at this. This really is a language. Now, cells they're not aware that they're using this kind of language, but we can think of it as a language, that the triplets stored in DNA specify words that we would say are the amino acids. So three-letter triplets specify the amino acids, and that's what we call the genetic code.